The actual anesthetic implications of protein receptors is that many of our anesthetics don't really they may actually go into the lipid bilayer, but they actually cause the effect by affecting the proteins. Okay? Most of the anesthetic agents that we have will actually bind specifically to a single type of protein receptor. So when we're talking about, let's say, a neuromuscular blocker, we're talking about a specific protein, which we have a name for it. In this case, we call it acetylcholine nicotinic receptor, that we actually bind to and shut off so it doesn't work anymore. And then as a consequence, the muscle will not contract. There's other forms of proteins, which we call receptors, where the anesthetics actually work on. Okay? Now, some of these can actually work by activation. Sometimes they can actually work by inhibiting. Sometimes they can also affect the secondary messenger activation. And they can act as a competitive or a non-competitive agent. Most of the time, we want our agents to be competitive, okay, because that means that they're reversible. Okay? Non-competitive agents cannot be reversed. It will depend on how long the molecule stays there, depending on protein binding capacity for the duration. Okay? The difference between an anesthetic and a poison is that the poison doesn't come off once it binds to the protein. It becomes irreversible, versus the anesthetic will reverse over time. Okay? Receptors are found in specific cells and organs, so you have to understand that too. Being able to selectively block or interact with receptors leads to many effects of medications, which is a lot of times the detrimental side effects are binding to other receptors that are not associated with the desired effect. We're going to be talking about this a lot in pharmacology, which we call pharmacodynamics. Okay? And I'll try to give you that as well in the MindJet map to sort of help you, if you work in pharmacology, to sort of develop little MindJet maps when you're taking care of medicines. And if you started doing that, I think you'll become a better clinician as well. I do that as already. Intracellular proteins can also be affected by different anesthetics because they actually will transduce or to get all the way through the lipid membrane and actually affect intracellular proteins. Now let's go on to carbohydrates. Carbohydrates in the membranes are not as important as the other components, but they're important for intracellular recognition. It's what differentiates you from being who you are in comparison to somebody else. Okay? Some parasites will actually steal the carbohydrate surface of your lipid membrane and cloak themselves in it. Okay? As a consequence, the body cannot identify the parasite from itself. Okay? So it can't attack it. But really, the body uses the carbohydrates or glycoproteins as a major identifier of enemy from self. Okay? If you're looking at autoimmune diseases or disease processes where the glycoprotein is actually altered or the sensing mechanism of self has been altered, and now the body begins to attack itself, causing damage in the long term. The other important component, of course, is carbohydrate. Is glycoproteins is the ABO blood group, which we talked about before. The other important structures or proteins are cell-to-cell adhesion. You got to put cells together, right? I mean, if you put 10 cells together and there's no way to bind them, then they just sort of form a glob. It would be goo. Okay? Instead, there's actually important structural components that attach the cells to them. If you look at anything that lines any kind of tubing, so you're looking at blood vessel, GI tract, okay, lymphatics, anything that is made out into a tube, the first layer before the cell is attached to it, facing towards the lumen of the tube, is what we call a basement membrane. Okay? That basement membrane is basically an attachment point for the cells. If you look at the GI tract, just think about that analogy. Okay, you got a basement membrane, and on top of that is your cells. And then the lumen is in there. That's where the food's coming through. Okay? So when the food is taken up, the cell on that basement membrane has to pull the food out of the lumen and then push it through the basement membrane so it gets into your bloodstream. Okay? So the basement membrane is one of those important structural components that don't really require a lot of oxygen to keep it going. Okay? It is deposited by the cells, but it is a basic structural component that holds the cell in place. If you disrupt the cell, in other words, you kill the cell, the basement membrane usually stays intact. Okay? Now, if you're looking at a blood vessel, the same kind of thing. The blood vessel has a lumen and with basically cells that are specialized that are inside that, and those are called endothelial cells. Okay? The reason endothelial cells are so important in our job is that endothelial cells, we used to be just thought, well, they're just the lining of the blood vessel, and that's all they did. And that's what I was taught when I was first started. Actually, what we found out is recently, in the last 10 years, is that the endothelial cells actually have a very important component. They produce basically molecules that prevent clotting from occurring. So proper endothelial health keeps the blood vessels from getting clot development. Okay? If you damage that cell, the basement membrane will be exposed, which then causes the clotting cascade to be activated. We call that the intrinsic system, okay? Because it's within the basement membrane. So the intrinsic system will activate, which activates to a certain degree slowly, but it still activates to form what's called fibrinogen, okay, which forms a clot. Okay? That's the intrinsic system. If you penetrate the actual basement membrane, and you're now exposing the next layer in, which contains collagen a lot of times, that will be the extrinsic. It's now exterior to the basement membrane. Now, which one's a more bad effect, you think? A cellular denudation or a basement membrane exposure and fracture point? That's much more serious. The basement membrane actually being damaged and being opened is a much more serious injury. As a consequence, the extrinsic mechanism, when it kicks up, gives you a much higher percentage of production of fibrinogen. Okay, so it'll clot the cell, that blood vessel very, very fast in comparison to the intrinsic system. Okay? Both are very important depending on what they do, and, but they are ultimately controlled by the endothelial lining and exposure to the basement membrane. Okay? The basement membrane is produced by fibroblasts, which are specialized cells that produce this stuff. It contains different kinds of collagen in it, okay, which is usually type 4 collagen. It has proteoglycans and plasmin in it. It's an interwoven matrix that doesn't have any real pattern to it, but it does interweave, and it's a very, very strong interweave usually. Okay? The cells adhere to it, and the cells then also talk to each other sometimes by forming protein connections between each other. Okay? These protein receptors can either be open, where the cell talks to one cell to the next. The typical example of that is in the heart. Okay? The heart muscle cells, okay, are individual cells, but they literally talk to each other from one cell to the next. The electrical activity that's generated by a pacemaker cell propagates into the myocardial tissue and dissipates from one cell to the next without having to have any kind of axonal control mechanisms. It's just simply because the protein channels are connected between one cell to the next. Skeletal cells don't have that. Okay? Skeletal cells, the only way to activate them is by activating them at the neuromuscular junction. Okay, so if you see one myofibril contract, if the other myofibril does not have an axon bound to it, it will not talk to it and it will not contract. Okay, so one system is set up so you can actually gradate how well your contraction is, versus the heart is based on how much energy propagation I'm generating. If I get a big enough signal, the whole heart fires at a greater intensity. Okay? Versus skeletal muscle, you can get a big firing in one myofiber and it will not matter to the rest of the cells because they're not gap junction, they're not talking to each other via what's called a syncytium. Cell receptor junctions can be called desmosomes, which are basically adhering junctions. Tight junctions can also be called impermeable junctions, and the gap junctions, which I was just talking about, which are adhering junctions, which allow for the electrolyte activity as well as electrical activity to jump over from one cell to the next. Depending on what cell you're trying to talk about, they have different capabilities. Now, the anesthetic implications of cell junctions is very important in the sense that it tells you how well capillaries will stay intact. Okay? If you look at most capillaries in your bloodstream, they're tight junctioned. If you take your hand, this is a clinical analogy, is you put your capillaries, they have podocytes, which are cells that are feet that touch each other. Okay? Capillaries and most blood vessels in your body are extremely tight junctioned. Okay? So they're tight like this. You can actually pick up water and hold it with your hands like this. If you go into septic shock, what happens is that these gap junctions or tight junctions that are holding these blood vessels together get attacked, okay, either by the immune system or by the bacterial lysis effects. Okay? And then what happens is the gap junction opens, and then now you have, instead of a tight junction, now you have like a colander effect, and the water will pour out of you. Okay? You guys have all seen this in the ICU. When you take care of somebody who's in septic shock, you give all this fluid, and it doesn't seem to stay in. And it's simply because the actual gap junctions and tight junctions have been injured, and now you can't keep the fluid inside. Okay? Cell junctions are also important in different kinds of structures, like the heart. Pacemaker cells actually generate the electrical activity, which then propagates into the myocardial cells, which do the contractional force. But the propagation doesn't go from one cell directly via this electron, via like an axonal system where it actually binds and causes a receptor. Instead, that propagation of electrical signal goes through these gap junctions, which we call syncytial cells. Syncytium, sorry, which is the whole system is talking to one cell to the next. These cell junctions can be affected by many disease processes. When we talk about pathophysiology next semester, we'll go into that some more. And different exposure to the base membrane has different effects on the coagulation cascade, which I talked to you about, the difference between intrinsic and extrinsic mechanisms a second ago. These cellular receptors are specialized cells as well. These are proteins, okay, that recognize and bind to chemicals called ligands. Okay. When we're talking about pharmacology, the way you have to really understand it, most of our drugs affect proteins in one way or another. Okay? If we're talking about a water-soluble compound, we're talking about it just attaching to a lipid membrane on the outside of the protein. Okay? So it does not go through the cell if it's water-soluble. If it's a lipid-soluble drug that we're talking about, then it usually has an intracellular mechanism of action. Okay? When you bind to, if you send a signal to a receptor, when that signal binds to that receptor, you can get an activation or inhibition depending on what kind of receptor it is. Okay? So, and the greater the number of signals that you have, which means transmitting signals, and you have enough receptors, and you get an amplification or greater effect. Okay? Now, what the receptor does is it not only just tells the cell, hey, look, I just got activated, but it does something rather interesting. It amplifies a signal. Okay? So for every one receptor binding, you get 100 signals produced as a secondary messengers. Okay? So it amplifies the signal. Okay? The secondary messenger, if you didn't have that amplification taking place, then the cell would not do what the receptor is trying to activate. Okay? So you can have different diseases where the secondary messenger is depleted and you don't get the effect, even though your receptor is binding. Let's talk about what would be an example of that. Okay? You have a situation where you take a patient in the operating room and the person begins to hemorrhage massively. During the process of hemorrhaging massively, you give 4 to 8 units of packed RBCs. And then you notice after that even though you've given adequate volumes, that your blood pressure doesn't seem to be coming up. Okay? You give ephedrine, and again, you don't see any effect. Okay? Ephedrine, or let's say we're using neosynephrine, which is a direct-acting agent. If you give neosynephrine to somebody, it goes to the receptor, and then it causes the secondary messenger to be released, which then causes the muscle to contract, which is smooth muscle. Okay? That secondary messenger, which you're going to find out is very important, which anything that wants to do contraction, what's the secondary messenger? If you're going to do muscle contraction, what's the activator of muscle contraction? Calcium. If you want anything to get anything done for workload, you have to have calcium. Okay? So calcium is one of the essential secondary messengers that you have. Now, what I just gave you the clinical scenario is you're giving a drug that's a direct receptor agonist and you're not getting any response. The effect is basically the secondary messenger somehow is depleted. And I told you the secondary messenger is calcium. Okay? Why would it be depleted? Okay? Yeah, what does that have to do with it? How does it do that? Okay, we're good. But she's got it right. I'm actually very happy to hear that. She's got it right. So the major source of the hypocalcemia is the blood transfusion. Okay? If the patient was transfused with 8 units of blood, usually at about 4 units of blood, we start getting toxicity from the blood component. Okay? What is inside packed RBCs? Citrate. Okay? Citrate. Citrate is made to bind to calcium so it scrubs it out so it doesn't cause the blood clot to form inside your packed RBCs. Okay? So when you give a large enough dose of it, it begins to scrub all the free ionized calcium in your bloodstream, and it begins to extract it out of your cells as well. Because to replete the utilization of calcium, you must bring it in from the outside source as well. So if you deplete the outside source, you eventually deplete the inside source. So all you have to do to reverse this effect is just give 500 to a gram of calcium, and all of a sudden the blood pressure returns back to normal. Okay? Again, this is why we teach you this stuff, so things don't just say, oh, well, I just got to memorize it. No. If you memorize it 6 months from now or a year from now when you have to go to clinical, you're not going to remember this. Okay? You need to start putting all this stuff together. Okay. Now, cellular communication can occur via cell-to-cell or via distance communication or distance education. Plasma membrane receptors are classified via signal transduction, which means they can be ion channel linked. The typical example of ion channel link would be muscle in the heart. Those are ion channel linked. You can also have catalytic or enzyme linked plasma receptors. And then you have the secondary proteins, which we call the G protein linked, which is very important in the sense that most of our molecules that are receptors are somehow linked to a secondary messenger system, which we call the G protein linked channels. This has really become important in the last 5 or 6 years. I still don't teach it to you guys in incredible detail. In the DNAP, if you're interested, then we do go into this for about 2 weeks. So it shows you how much time you have to spend on it. And there's not enough time with what else I got to teach you. The extracellular messengers convey instructions to the cell's interior. The first messenger is usually the extracellular ligand. This is the stuff that's floating outside the cell, and it comes into the door, which is a protein, and it causes it to have an effect. The analogy would be like you have somebody that's outside of this room. Okay? They would come to this door, and instead of trying to come in and say, hey, how you doing, the way they would actually convey the information is they would hit the door and knock on it. The secondary messenger at that point is the sound waves that comes from the door noise to me. Okay? In this case, the secondary receptor would be something like calcium or some other kind of protein that's being synthesized, or even a lipid that's being synthesized from the effect on the actual protein. Okay? So the first messenger is outside the cell, okay, but it transfers the signal to the intracellular messenger, usually via secondary messengers, which passes on the signal. It triggers a cascade of biochemical events, which can be very complicated, which I'm not going to go into a lot of detail on that. And the two main pathways are cyclic AMP and calcium. That you have to know. Okay? The two major pathways that we use is cyclic AMP and calcium. I can go into, there's 25 of them or more, okay? But I'm just going to get you to understand 2 for now, okay? Because that's the ones that we use most of the time. If we're talking about calcium, we're talking about usually something that's having to contract. Okay, smooth muscle, skeletal muscle, a cardiac muscle, all those are calcium dependent. Okay? If I want a cell to do something else, okay, then we can use other secondary messengers like cyclic AMP. Cyclic AMP will actually increase contraction velocity. Okay, so not only do I sometimes use calcium to increase contractility, but I can also use secondary messengers like cyclic AMP to increase my inotropy or chronotropy of the heart rate. Okay? The typical example of this and why we come into this is when we talk about phosphodiesterase inhibitors. Cyclic AMP, once it goes into your into your cell, it gets broken down to cyclase and I think adenine or cyclase, I can't remember right now, but it breaks down. Okay. In the process of breaking down, okay, it basically the secondary messenger becomes depleted. We can give what's called a phosphodiesterase inhibitor that prevents the breaking down of the cyclic AMP and basically drives that secondary messenger levels up, which then gets the desired effect of increasing contractility. So your drugs like Inocor, the way they work is not at this receptor level, they work intracellularly by preventing the inhibition of the breakdown of cyclic AMP. Now, the different cells can do different things. They can send signals across gap junctions, which is what the myocardial cells do. You can get transient direct link up of cells. We don't, the human body doesn't do that very often. Other cells do that in free environments. You have paracrine secretions that can take place. We'll talk about endocrine secretion as well, which is hormonal secretion, where the receptor, the signal is sent through the bloodstream, which we call a hormone. And then the hormone has a long-distance effect on a target cell. And there's different forms of hormones that can either penetrate the cell membrane or stay on the surface. You can also have neurohormonal systems, which we'll talk about, which is a part of the posterior pituitary. And we'll go into these different ones more specifically in time. Now, next portion of the discussion is membrane transport. Okay? To make a cell alive, you have to make the cell inside different than the outside. Okay? So my cellular environment in my body, I want to keep very stable. Okay? I don't want to have large fluctuations in pH in my cell. I don't want to have large fluctuations in my electrolytes in my cell. I want to keep things very steady so the cells are happy. Okay? And that's basically what we're doing when we take care of patients. We're changing your extracellular content, but we're really trying to keep the intracellular milieu pretty much the same. When we start messing with the extracellular space, we eventually affect the intracellular space with time, which can be very deleterious. Okay? The molecules that you have to move in and out, of course, are water, ions, which are electrolytes, metals, which are rare, but they're still essential, vitamins, sugars, fatty acids, and amino acids. Okay? And it depends on if you want to use passive or active transport mechanisms. Active transport means that you have to use energy to transfer the molecule. Passive means that you use gradients to let the molecules transfer across. Many of these gradients are established by active processes at times, but most of the gradients that we're talking about at times will be passive. First part, electrolytes. Electrolytes have electrically charged and dissociate into what's called cations and anions. Okay? What we measure that is in milliequivalents per liter, okay? Which is electrical charge per unit volume. Okay? And that's tricky for most students. It's not milligrams, it's milliequivalents per liter. If I tell you that I have 1 milliequivalent of sodium, okay, that's let's say that's just one little molecule of sodium. Okay? If I were to have 1 molecule of calcium, how many milliequivalents would be calcium? Would it be 1 milliequivalent? It's 2 milliequivalents, because calcium always comes in a 2+ charge. So even though it's 1 molecule, it's 2 milliequivalents per liter. Okay, that's the charge component. So it's very different than just a molecule now. You're talking about not millimoles or anything like that. You're talking about milliequivalents per liter. Okay? We tend to use milliequivalents per liter as a discussion when we give electrolytes to fluids. If you look at all our IV bags, they're in milliequivalents per liter. Okay? So make sure you understand that. The reason we're doing that is because we're trying to apply what's called Nernst equation on everything, which means that one side of any compartment must have what charge? So if I had to say, okay, how many people are in this room right now? There's 54, right? There's 54 people in this room. How many people are outside this room? 0. Okay? But if I were in this room, half the room is positive and half the room is negative. So the total charge state in this room is 0. Okay, so in this compartment, it's 0. If you look at the outside compartment, it's 0 also. But if I open that door and I tell you, hey, day's over, where do you think where most of you are going to go? Out the door. That's called a gradient. Okay? So even though there's 0 charge in here, you still have the potential of doing work because this compartment is different than that compartment. Okay? You're able to transfer across. Okay? Non-electrolytes basically do not dissociate, so things that don't have a charge. Most transport mechanisms to a certain degree have passive components to it. Okay? In other words, require energy, which of course are the active. Okay? A typical active component would be the sodium-potassium ATP pump. That's the first really protein pump that was really well described. For every 3 molecules of sodium coming out, you only pull 2 of potassium back in, and that requires ATP for that to actually take place. Now, passive transport is possible because you have a semi-permeable membrane. Okay? If you have an impermeable membrane, guess what happens? Nothing. Nothing gets across it. Okay? If you had a permeable membrane, then everything gets across it. Okay? So we have to have what's called a semi-permeable membrane. What makes a membrane semi-permeable is the proteins that are embedded in it, especially the transmembrane proteins. How I control the traffic between one portion of the cell to the other is very critical for this concept. Okay? Now, you can actually get semi-permeable membranes to take place also in non-living tissue. Okay? So you don't necessarily need the proteins for that to take place. And the 3 mechanisms that really, when you're talking about semi-permeable membranes, are diffusion, hydrostatic pressure, and osmosis. Okay? Now, let's go into the specifics. Why am I getting you again? This is basic science, I know, and you're going like, why do I have to know this? Okay? All right. Let's talk about some basic concepts first. Let's say you have somebody who's hypertensive. Okay? Somebody who has high blood pressure. Let's do a little class and discussion and find out how many people, if you have a hypertensive patient, how many of you believe that that person has a very high water content in their bloodstream? How many people would say that the person has a very high water content in their bloodstream? Okay. How many people say that the water content is very, very low? Okay. Why would you think it's very low? Okay. Here's how it works. Okay. You have a membrane. Okay, and that membrane is under certain effects. One is hydrostatic pressure, which is how much pressure is being inside, actually will push the fluid through. Okay, so you have somebody who's hypertensive, the water inside that blood vessel will actually get squeezed out. So most people that are hypertensive are actually hypovolemic. And you put them to sleep, you find out real fast if that's the case. Okay, they'll actually cardiovascular collapse on you when you put them under anesthesia. Okay? So hydrostatic pressure is very critical for how much volume will be displaced out of a cell, okay, or out of a lumen of a blood vessel. We'll talk about diffusion here in a minute, and then I'll go into osmosis. Okay? Diffusion is basically passive. It basically follows the law of Fick's law. Diffusion is what we use in anesthesia constantly, okay, in the sense that it's a discussion about how anesthetic gases come in and out of you. Okay? If I give you an anesthetic gas and the concentration outside in the alveoli is, say 50% of nitrous, and you have 0 inside the bloodstream, that's considered a gradient. Okay? If I gave you 60 or 80% nitrous, would that mean that does that make a bigger gradient? Let me reiterate that. Okay? You're inside of a membrane. Okay? You're inside of an alveoli. Okay? You have the capillary wall is on the other side, and you're inside the alveoli. Okay? There's 50 of your partners, and your 50 of your buddies want to get across into the blood vessel, which has none of you. Okay? Now, if you take the same alveoli and now put 80 of you in it, which one is more likely to get across? The 80 or 50? to 80. Okay? The greater the concentration, the more pressure can be exerted by those molecules to get across. That's what we call a concentration gradient. Okay? That's what we establish constantly in anesthesia. When we turn on the gases onto the patient, we establish a high gradient concentration of gas, which then gets into the bloodstream. If I want to reverse the effect, I have to shut down the gradient, right? So the way I do that is by giving high flows of oxygen to the patient. The oxygen goes in there, extracts all the nitrous, so we're down to 100% oxygen inside of the alveoli. And now the 80% that's inside the bloodstream now comes pouring back out. So it's completely what? Passive. It's totally dependent on concentration gradients. So that's why we teach you this stuff, so you begin to understand the simple concept of how an anesthetic gets into the body and how it gets out. If you don't allow the person to ventilate properly after the case, okay, the person's anesthetic will not wear off. Okay, if you don't flush the system out and get all the content out of the alveoli, the gradient won't be established and you won't get the gas out. The person will still stay asleep for long periods of time. Okay? The gradient can actually be very slow to come out too, and you can smell the anesthetic gas for 3 to 4 hours after surgery. If you work in recovery room, you just go by somebody's face and just swipe with your finger, like my hand like that. You actually will pull air towards you from the person's breathing out, and you'll smell the gas in them. Okay? So it takes sometimes a long time for all this stuff to come out. So, but passive diffusion is completely passive. It's basically dependent on concentration gradients. Okay? The other important part, which is Graham's law, which is inversely proportional to the square root of the molecular weight. Again, you can memorize that statement or just think of it basically. What's easier to walk through this door? A human being or an elephant? Right, and something small gets through easy. Okay? So keep it simple in your mind. The bigger the molecule, the less it's going to travel through the membrane. Okay? So it's inversely related to the molecular weight. So if the molecular weight goes up, which is basically talking about size, then you're less likely to get through. So diffusion gradients are dependent on concentration, molecular size. The other thing that's important is ionic charge. The more positive, the more negative charge, the less likely it's going to get through. Lipid solubility is also important. Believe it or not, most of our good anesthetics are insoluble. You would think, we want something that's really soluble. The problem with something very soluble is it comes in really fast, and then what does it do? It doesn't want to leave. Okay? So it's a great anesthetic, but it just won't quit. Okay? So the example, that's ether. You give somebody chloroform and ether, it works fantastic. The only problem is it's going to work for days. Okay? Versus desferrine, which is very insoluble, will only work for a few minutes once you shut it off. Big difference. Titrate it in, get it in. It's hard to get it in, but once you get it in, it gets a good effect. And then when you want it out, it comes out fast. Okay, so it's dependent on, of course, lipid solubility. Membrane thickness as well. Okay, if you have the actual alveolar membrane, or whatever the membrane you're talking about, if it becomes thicker, it's less likely to get across. And of course, your membrane surface area. The greater the surface area, the more likely it is to get across. The less surface area, the less so. Now, hydrostatic pressure is a mechanical force of water against the cell membrane. Okay, it's a counterbalance of osmotic pressure, which is to a certain degree is what the molecules that are inside the blood vessel. Okay, so basically you're looking at we just talked about that analogy of hydrostatic pressure, which is high blood pressure, will actually push fluid out into the interstitial space. Osmosis is water movement down a concentration gradient. Okay, the simple example of osmosis is a simple statement: is water goes where the salt is. Okay, the analogy to this, or the everyday life experiment that you guys do, is that we're coming up to Valentine's Day, and you better have some roses or flowers for your significant other. Okay, but one of the things you can do is to do one or two things. You can put your plant in water. When you put your plant in water, what you'll see is the next day that the actual water level drops. Okay, so where did the water go? Water went from a zero concentration of electrolytes to a area of high electrolytes, which is inside the plant cells. Okay, if you want to see that plant that flower sort of die overnight, take a bunch of salt, pour it into the water, make it salty water, and then put the flower in it. The water will come out of the plant, and the plant will die overnight. Not something smart to do, okay? But, just something to sort of show your kids so they understand what osmosis is, okay? So water goes where the salt is, okay? The reason this is important is because we deliver what we call isotonic solutions to most of the time, which means that we're trying to give a fluid that's not going to displace into or out of the cells, okay? If we give somebody normal saline, that tends to be a little bit on the hypertonic side, okay? Normal saline has 154 milliequivalents per liter of sodium in it, okay, which makes it hypertonic in comparison to your normal, which is 140. So as a consequence, water gets pulled into the blood vessel, but it's getting it from somewhere else. It's stealing it from the interstitial space. If you give D5, normal saline, that's still a hypertonic solution. D5 is basically just sugar, and it'll get rapidly uptaken by the cells. If you give D5W to somebody, okay, the person will survive, and because it's a little bit more on the hypertonic side, but if you were to give just somebody straight water, you're likely to kill them, okay, if you give it to them IV-wise. The reason that's such an issue is because if you give a severely hypotonic solution, water goes where the salt is. Where's it going to go? It's going to go into the cells very rapidly, okay? And the major important cells that are in your bloodstream are what? Your trucks, your red blood cells, okay? So if you give somebody a lot of, just free water, the red blood cells will just expand really fast and explode, okay, killing that red blood cell, okay? I've seen an example of somebody giving, by accident, hypotonic water, which is straight water, into the brain. Not a good day, okay? That's something you don't want to see. That's why most ORs now have taken away all the free water. They used to have that to debride tissue, but it's extremely dangerous if you're working in the neurosurgical OR. If they put free water onto the brain, it's not going to be a happy brain for very long, okay? Water goes into the cells and causes them to burst. Okay. On the other hand, hypertonic solutions, we're using that in the military to basically you give a slow amount, very limited volume. And water leaves the cells, causing crenation, or cells to end up shrinking. So this is the example that we use in the book. Okay. This shows you, I'm not going to go into a lot of detail, this shows you before and after addition of osmotic disequilibrium, and eventually shows you that the thing eventually equilibrates across the board to be equivalent in both compartments. And that's what you need to understand this concept. Then you have the next concept, which is mediated transport. Mediated transport can either be active or passive. A transmembrane protein that is selective of what it will transport. It's much faster than simple diffusion, but it's saturatable, okay, which means that if I give enough of the molecule, it'll stop moving, okay? It'll stop going at a certain rate. If I ask you all to run out one single door, the first 10 might get to the door, but after that it becomes to slow down because there's too many of you. You're saturating the door quantity. That's what it means. Okay? Competitive inhibitors can be part of the mediated transport. They bind to the transport site and compete with the transported molecule. Things like this would be neuromuscular blockers are competitive inhibitors. Okay? Non-competitive inhibitors bind somewhere else portion of the receptor that not to the, and it basically causes almost irreversible effects. If it's a poison, it's completely irreversible. If it's a prolonged acting agent, it can reversibly bind, but it's going to have a prolonged effect. These are your actual proteins that you're looking at. There can be different kinds of proteins that are transporters. You can have a uniport, which is a single molecule, goes across one side to the other. This is a simple gradient usually at times, but what limits the transfer across is depending on how the actual molecule configures and opens during a certain time period. Some of them are voltage gated, and some of them are calcium gated, depending on the situation you're talking about. You have also your symport system, which means that you're bringing in two molecules together, usually one down a gradient, and then the other one uses that energy to transfer the other molecule, macromolecule, with it. Typical example of that would be insulin. Okay? Insulin drives sugar in it as well as potassium in it together. So that's an insulin symport system. The antiport system, on the other hand, is a system where you have one gradient going in the opposite direction and pulls the other one in, usually against a gradient. Types of mediated transport can be what's called the ping pong theory, which is the sodium-potassium ATP pump. The sodium-potassium ATP pump is basically a configurational change in the protein to allow for 3 sodiums to come in from the outside, 3 sodiums from inside the cell to come out, and 2 potassiums to come into the cell. Now the reason that's the difference is, I'll show you here in a second in the picture, but I'll go back to it. There are also channels, uniport, symport, antiport, which I just showed you before. Now when you go into active transport, this really requires life, and it requires energy. Okay, so you're looking at ATP as your main power source. You're transporting large molecules or charged molecules across the membrane. So you're looking at a lot of workload to do this. The mechanism uses the energy generated by cellular metabolism, usually oxidative phosphorylation under most conditions. So you're using mitochondria. Sometimes the cell can use non-oxidative mechanisms, which we call fermentation, or anaerobic mechanisms, which is something that our cells don't do well with. So we usually don't survive very long with that kind of situation. These are receptor-mediated. They can actually are active transport pumps. Usually the ATP is the main power source, and sometimes the sodium-driven gradient can also be helpful. Okay? Because remember I said to you that ATP pumps basically pour a lot of sodium outside the cell, right? So now sodium is in very high concentrations outside the cell and in very low concentrations inside the cell. So when we open up a protein channel and we allow the sodium to come across, then it establishes a gradient, and that creates energy, and when that can do work. Now you have to ask yourself, why does the body use sodium? Why not use potassium? There you go. Very good. Sodium is a smaller molecule. Okay? The way you want to look at it, if you want to look at size equivalency, the easier way to describe it is think of a sodium molecule as a hard ball and a potassium molecule a softball. Okay? So if you can actually you can actually only selectively pick out that ion simply by making a channel small enough that it doesn't allow potassium to go out. Okay? If I make the channel just big enough for a hard ball, then that's the only molecule that's going to come through. Okay? And most molecules are bigger than sodium. Okay? So usually that's the reason for that, why the cell picks on that. Sodium-potassium ATP pump is this mechanism where it takes 3 sodiums inside the cell and puts it on the outside. And then it takes up 2 potassiums and brings it inside. This requires ATP to do this. This is a more fancy picture showing you the actual mechanism, which is requiring ATP down here to actually function. So you have your ATP molecule that actually generates the energy to cause a configurational change of then expressing the potassium out. But what this picture doesn't do a very good job of is it doesn't explain to you how potassium is a much bigger molecule. And when you look at this from this side, this is molecular weight. Remember your basic chemistry? This is why we tell you to take basic chemistry to a certain degree so you can begin to understand these concepts. If you look at sodium, the molecular weight is 11. Okay? Look at potassium. Potassium is almost twice the size. It's at 19. So that molecular weight is actually physically means that molecule is bigger. So if you were to have a catcher's mitt and you're able to hold 3 hard balls in it, okay, but when you throw the 3 hard balls out, okay, and you have to want to accept 2 more molecules that are the size of softballs, that's all you can put in your glove. And that's what you pull back in. So it's a size limitation to the protein. And that's as simple as that. Okay? So hopefully that'll, if you take a board exam and they ask you a question like that, you may not remember 3:2 ratio, but you may at least remember that, okay, Dr. Nolan talked about hardball and softball, and you'll keep that in mind, and you'll know that sodium is smaller, and you get that 3:2 exchange rate. Now there's also active transports that occur at the cells that are via vesicle formation. These are active transport and requires usually a large movement of molecules, and this can be called by endocytosis, which is the actual involution of the plasma membrane. Okay? If it's just simply for water drinking, it's called pinocytosis. Pinocytosis is basically just the cell grabbing water from the outside environment and pulling it inside of it. It also can do things like phagocytosis, which means it uptakes like bacteria or large molecules. The typical example of phagocytosis is usually seen with white cells. You can actually see that under microscopy. And then you can also have what's called receptor-mediated endocytosis, which the receptor then binds to whatever it's trying to bind and pulls that molecule in. Exocytosis is reversing the process where you actually dump stuff out. Hormones are it's a certain way exocytosis. So this is a picture of, let's say, a particle that's being pulled back in. This is endocytosis. Once it pulls this molecule in, it forms a vesicle around it. And then what eventually causes the digestion of this to take place is for lysosomal sacs to bind to this, which is the digestive component of the cellular organelles, which then breaks down and hopefully destroys this compound and then utilizes those products as final byproducts for something else in the cell. Whatever the cell cannot digest, it forms into a membrane-bound vesicle. Some disease processes where you can't get rid of it, it begins to just accumulate in the cells. And that's very bad. It eventually causes bad effects to the cell. If the cell can't get rid of it, if it can get rid of it, it'll dump it into the interstitial space, which is why lymphatics are very clearly important because they scrub out this junk. Okay? So lymphatics are clearly important for cleaning out cellular debris that's produced as cellular metabolism. The cytoplasm is the next structure you think of. This is a fluid that's inside the plasma membrane. Okay? It's a cytoplasmic matrix, which we call the soup of life. Okay? The reason it's called the soup of life is it's got all the essential enzymes for most of the essential pathways, biochemical pathways. So gluconeogenesis, glycolysis, breaking down of glucose and producing energy, all those things are found in the cytosol. There's literally thousands of enzymes that are found inside the cytosol. And trust me, we have no clue how everything works yet. Okay? We might think that we understand it, but we really don't. Okay? I used to be a chemist in my past life, and we used to talk about all this stuff, and there's a lot of stuff that we don't understand yet. Okay? What's important about this is that these enzymes are proteins, which means that they're being generated by the cell in a certain way. Okay? The way the proteins are generated is that they're taking a messenger RNA, they're translating it, okay, into a protein using what's called a ribosome if it's inside the cytoplasm. Ribosomes are just a specialized protein factory that helps produce other types of proteins. And we'll stop here and start back up in 10 minutes.